Hello, everyone. Uh, yeah, hope you received my message. So actually, we changed the schedule. Um, yeah, because some scheduling conflict. Professor Tim Park will join us on Wednesday also. So each guest lecture will be like 1 to 1.5 hours. So in our first part of lecture on Wednesday, we will have Professor Tim Park, and for the second part, we will have Professor Shlomo Dubnov as our guest. Okay, so um, so that means today will be our last kind of main lecture that I will give you. Okay, so before that, actually I added another playlist here. Um, yeah, I know it's a little bit late, but if you are interested in mathematical music theory, you can check this out. Okay, we will also cover a little bit music tuning in our lecture today. Okay, not really much because we don't have time, but if you're interested, there will also be some, books, references, like here, like this book, also covers some, basic math of pitch, scale, and harmony, yeah, related to music. And another thing is that, yeah, our TTSM workshop is up for this year, this summer, and I will teach a workshop there this weekend. Okay, so Saturday and Sunday. Okay, so this is what I'm going to teach. So each workshop will be kind of 4 hours. So yeah, it will be 8 a.m. Yeah, it's a little bit early because I have something scheduled for, the afternoon Saturday. So I will do like 8 to 12 in the morning on Saturday. And on Sunday, it will be 10 a.m. to 2 p.m. Okay, so it's a very intense workshop for one weekend. And if you want to, you can just register there. Let me see. Yeah, this is this enroll. Yeah, you can choose which one you want. And this is taught by my colleague, Torniki. So I will talk about the symbolic music generation using neural networks. So basically, recurrent neural network, things like that. And he will talk about audio, audio kind of sound synthesis and music generation. So I will talk about like more based on symbolic music in the MIDI format, and he will talk about the audio format. Okay, like what we just covered, the waveform, frequency, sampling, things like that. So it will be different kind of focus, but we will all use this neural network. I don't know what he will use because he used to work with VAE, variation autoencoder. But this year, I think he kind of shifted to this diffusion model, which is, a hotter topic. But I don't know if he will really talk about, diffusion model in his workshop or, but yeah, this is the basic kind of information and the plan, and he will teach the next weekend. Okay, so we only have 2 workshops this year, both on technology and machine learning, artificial intelligence. Yeah, and this workshop is free. As long as you have time, you can just register, and yeah, we will just let you in, and you can join the Zoom meeting like what we are doing now. Yeah, and have some just, if you're interested, just have some other knowledge. And we will have international participants, not only from US, and we will probably have like someone from Mexican, someone from Japan, also like a lot of people from Russia, because our organizer, yeah, he is Russian and he has this connection, with Russia. When I gave the workshop last year, like most people are from, this Europe area. So if you also want to meet some new friends, like international people, yeah, and this will be a good chance. Like we will also have this group chat on Telegram that people can, we have this group chat and you can just communicate and maybe you can just get to know some interesting people. Okay. Um, so let me know if you have any questions. But this is just some, something happening soon. And today, let's jump into our last topic. Okay, our last topic will be on psychoacoustics and music perception and something more like cognitive science, because we covered a lot of natural science, like sound waves, physics, like and also some math. So they are more like, I would say, mechanical. I mean, something like the natural law, right? Natural law of something. But what we cover today will also be some science. It won't be, something very subjective. It's also based on our auditory system, and how your brain your perception, right? Your processing, signal processing, the visual cues, the auditory cues, how your brain process this information. But this will be more like cognitive science. So it's different aspects. Okay. And also psychoacoustics, also including some kind of illusions, which is the discrepancy between the nature, right? The reality, and how you perceive these signals, right? How your brain understand those signals. So this will be a different focus, and maybe more interesting for some people are interested in, psychology, cognitive science, and neuroscience, etc. Okay, so let's just get started now. Slide show. Okay, so the first part will be on auditory process, or the ear physiology. So there's a lot of study, based on how your ear perceives sound, how you hear sound, and how it goes through your, auditory system, and finally how it's, reach your brain as an electrical signal. So we have some really good video visualization here that I can play for you. Okay.
Have you ever wondered how sounds make their way from the source all the way to your brain? Take a trumpet, for instance. When it's played, it makes sound waves in the air. The outer ear catches the waves, which then travel through a narrow passageway called the ear canal. The sound waves reach the eardrum, which is a membrane roughly half the size of a dime. They make the eardrum vibrate, which in turn vibrates 3 tiny bones called the malleus, incus, and stapes. These bones amplify or increase the sound vibrations and send them to the cochlea. The cochlea is shaped like a snail and is the size of a garden pea. It is filled with fluid, and the sound vibrations make this fluid ripple, which creates waves. Hair-like structures called stereocilia sit on top of hair cells and are grouped together as hair cell bundles inside the cochlea. The hair cells inside the cochlea ride these waves, and the hair bundles are moved. The hair bundle on top of the hair cell turns these movements into electrical signals. As the hair bundles are moved, ions rush into the top of the hair cells, causing the release of chemicals at the bottom of the hair cells. The chemicals bind to the auditory nerve cells and create an electrical signal, which travels along the auditory nerve to the brain. Different hair cells respond to different frequencies of sound. The hair cells at the base of the cochlea detect higher-pitched sounds, such as a piccolo or flute. The hair cells toward the top of the spiral detect progressively lower-pitched sounds, such as a trumpet or trombone. At the very top or apex of the spiral, the hair cells detect the lowest pitched sounds, such as a tuba. The auditory nerve carries the electrical signal to the brain, which interprets the messages as sounds that we recognize and understand.
Well, it's an interesting video that talking about how we hear sound and use different musical instruments to illustrate, right, the structure of cochlea and the frequency perception of the cochlea. Here is another one. Okay.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch, or frequency, produce a slower rate of vibration, and sounds of lower volume, or amplitude, produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth, known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the 2 fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
Okay, so this one is more professional, and it mentioned many terminology which I also not very familiar with. It's more like biological kind of related. And I think this video gives us a more kind of scientific or more professional illustration of the auditory system. Okay. And it's also like well-made, kind of fancy anime and good music. So here is I think this one probably give us a kind of different perspective of this theme. I personally like this most because for I'm not a neuroscience or cognitive science background. I don't have this background. And if you use so many terminology, it won't be very understandable for me. So this one actually interprets or explains this different frequency response are very clear. And I think that will give us a lot of intuition and a lot of prior knowledge when we get into our next topic on the auditory filters. And we also talked about fully transformed frequency domain, and actually our real auditory processing is related to this kind of process, which makes this fully transformed more like a scientific, right? It's not really only about engineering. It's not only like convenient for engineer. It also has this biological foundation, because this is really like how we perceive sound, right? We don't perceive sound in the time domain, although it transfers in this air or in this medium as the sound waves, right? But we don't perceive it in this waveform. Our auditory system, like this, actually transform this waveform to the frequency domain, and this is how we perceive different sound, different frequencies. So let me play this.
In order to distinguish between the sounds of a bass drum and something that has a much higher frequency, such as the sound of a bee's wings flapping in the air, your brain is relying on the cochlea in order to differentiate between the two different sounds. So the difference between a bass drum and a bee's wings flapping in the air is the frequency. So a bass drum has a very low frequency, whereas the wings of a bee when they're moving through the air very quickly, have a very high frequency. So as the information from a bass drum beating or a bee's wings flapping comes into the ear, they eventually hit the cochlea. And we went into a lot of detail about how exactly the sound wave is converted into a neural impulse by the cochlea that eventually reaches the brain. But now we're going to go into how the cochlea distinguishes between sounds of varying frequencies, and how this distinction is maintained all the way to the brain in order for the brain to be able to perceive different sounds. So this is known as auditory processing. Auditory processing. Your brain needs to be able to distinguish between sounds of varying frequencies, and you're actually able to hear things with a frequency of 20 hertz all the way up to a frequency of 20,000 hertz. So this is a huge range, and in order to distinguish between sounds of low and high frequencies, the brain uses the cochlea, and particularly something known as basilar tuning. So basilar tuning. And the term basilar comes from the basilar membrane, which is inside the cochlea. So inside the cochlea, there's actually a membrane that contains a bunch of hair cells. And if we were to unroll this cochlea, if we took the cochlea and we unrolled it, so it's normally rolled up like this. If we unrolled it, so that was flat, there are varying hair cells. So this would be the very base. This is the base of the cochlea, and this is the very apex, the very tip. So the base would be right here, the apex would be right here. And if we unrolled it and looked at which hair cells were activated given different sounds, we would notice that hair cells at the very base of the cochlea were actually activated by very high frequency sounds. And hair cells at the very apex of the cochlea are stimulated by very low frequency sounds. So let's look at another picture just to make things a little bit clearer. So this picture basically just shows the cochlea unrolled. And as I mentioned before, this would be the base of the cochlea. Let me use a darker color. This would be the base of the cochlea, and this would be the very tip or the apex of the cochlea. And hair cells are found all along the basilar membrane. So this membrane right here is the basilar membrane, and there are hair cells implanted inside of it. There are a whole bunch of these hair cells. And hair cells closer to the very base respond to a very high frequency, so this is 1600 hertz, and hair cells closer to the apex respond to a lower frequency, so 25 hertz. So this would be something like a bass drum, and something with a very high frequency would be something like bees' wings flapping in the air. So as sounds with varying frequencies reach the ear, they will stimulate different parts of the basilar membrane. So if we have a bass drum being played, has a pretty low frequency, and it'll eventually go into the ear, reach the cochlea, and it'll actually travel along this basilar membrane until it reaches the hair cell that is attuned to that particular frequency. So let's say that this is a frequency of 100 hertz, for example. The sound waves eventually cause fluid inside the cochlea to travel in such a way that the hair cells that are very sensitive to a frequency of 100 hertz, which looks like it's right around here, will actually activate. And these hair cells will fire an action potential. And this signal will eventually reach the brain, and it will be mapped to a very particular part of the brain. So this right here is the brain, and if you lift up this little piece of brain, there is something known as the primary auditory cortex. And the primary auditory cortex is this blue region over here, and it's basically responsible for receiving all of the information from the cochlea. And you can see that it's actually separated, similar to how the cochlea is separated to various frequencies. It's sensitive to various frequencies. This primary auditory cortex is also sensitive to sounds of various frequencies. So, for example, this would be a part of the cortex that receives information from hair cells that are sensitive to a frequency of 0.5 hertz. And this part of the auditory cortex receives information from hair cells that are sensitive to a frequency of 16 hertz. And the reason that this is important is because the brain needs to be able to distinguish between various sounds. So if we had all the hair cells sensitive to every single sound, then whenever you heard any sound, then all the hair cells would fire at once, and they would send this huge signal to the brain, and the brain wouldn't be able to distinguish between different sounds. So by having this basilar tuning, the brain is able to differentiate between sounds with a very high frequency and sounds with a very low frequency. And this mapping of sounds with a higher frequency versus sounds of a lower frequency is known as tonotypical mapping. And just to summarize, we have sound waves coming into the ear, and different sound waves have different frequencies. And we need to be able to distinguish between the different frequencies. So the sound waves come in, they hit the cochlea, and they will activate hair cells in different parts of the cochlea. So if it's a very high frequency sound, it'll activate a hair cell over here. If it's a very low frequency sound, it'll activate a hair cell over here. And these hair cells will actually send axons, and these axons eventually all bundle together to form the auditory nerve. And the auditory nerve carries axons from each hair cell inside the cochlea. And the auditory nerve eventually reaches the brain and will again separate its fibers and reach different parts of the brain.
Okay, so I guess this gives us a better illustration of the frequency response of the auditory system. Our next topic is on auditory filter. Okay, so our ability to separate the components of a complex sound depends, at least in part, on the frequency analysis that takes place on the basilar membrane. Actually, Fletcher and Helmholtz suggested that the peripheral auditory system behaves as if it contains a bank of bandpass filters with overlapping passbands. These filters are now called the auditory filters. Okay, so these kind of different a bank of bandpass filters are widely applied, in this digital signal processing engineering. But actually, they did this research and they kind of demonstrated our auditory system also has this kind of this auditory filter, which simulates this the structure of a bank of bandpass filters, okay, with overlapping passbands. And Fletcher thought that the basilar membrane provided the basis for the auditory filters. Each location on the basilar membrane responds to a limited range of frequencies, so each different point corresponds to a filter with a different center frequency. So when these bandpass filters have different center frequencies, makes our basilar membrane sensitive to different frequencies. Right, so different position corresponds to different frequencies, which help us to distinguish different sounds. So this is how this one filter looks like. Okay, so this filter, you can see it has this center frequency of like 450 hertz, something like this. So this is the center frequency. Okay, and this is the shape of this specific auditory filter. Okay, you can see it covers this range from like kind of 100 to like, I mean, this is really kind of a huge span. But I don't know if our real kind of every auditory filter has this kind of structure, but this is actually tested by this experiment called auditory masking. Okay. So the process by which the threshold of audibility for one sound is raised by the presence of another masking sound, right? So auditory masking means I'm playing one sound, like for example, I'm talking now, but now I'm playing some background music. So let's say a singer is sing a song, and when these two different sounds overlapping with each other, let's see if that singer is also has this female voice where she sings in the similar frequency as my speech, then my speech will be covered or be masked by her singing. Okay, but if this singing is kind of like of a very high or very low frequency, which doesn't really overlap with my talking voice, you probably will still hear my speech clearly and also identify another source. So this means that this kind of masking effect is frequency dependent. So when two sources have the same frequency, the maximal masking effect is achieved. Okay. So a signal is most easily masked by a sound having frequency components close to or the same as those of the signal. So in this graph, what you can tell is that so our masking signal, right, is at this frequency. I assume it should be 450 hertz, probably. And now, if we have a signal of the same frequency, okay, it will be masked mostly. So this kind of different curves here with 20, 30, 40, it is the loudness, okay, the dB, the loudness of the masking signals. So these different curves correspond to different levels of loudness, but in terms of the masking effect, always if I have another signal of the same frequency, or a signal maybe like 300 or 500, it will be very easily masked, okay? Which means I have to raise the loudness of my signal really high to make it, to make it audible. However, let's say we have a sound of 2000 hertz. Then if the loudness is, is below 80 dB, and then even if I'm playing some sound at 450 hertz, it won't really affect my perception of this sound at 2000 hertz, right? As long as it's below this 80 dB, because you can see for all those different curves below 80 dB, it doesn't really like overlap with this 2000 hertz, right? This the band limit of this bandpass filter we only like is restricted within a narrow band, right? Within a narrow band, it doesn't really extend to 2000 hertz. So it means if I'm playing like a sound at 550 hertz and another sound at 2000 hertz, as long as the loudness of this sound doesn't exceed 80 dB, I will hear both sounds clearly. It will be no kind of auditory masking kind of effect, even if I'm playing this sound really kind of loud. Yeah, as long as it's below 80 dB. However, if another sound at 500 hertz is played, then it will be easily masked by my another signal because they are close in frequency. This kind of masking effect is maximized, which means if I want to hear my signal at 500 hertz really clear, I need to raise the loudness, right, of my signal to make it exceed this masking signal, and then I can finally hear my sound. So this is the kind of effect, and this can be tested. Okay, so they did this experiment. They brought in some participants, and they played this one sound I hear. This is the masking signal, and they play different kind of signals to the participant, to the to their patient, I would say. And yeah, and you will, if you just join this experiment, and you will tell them, okay, I can hear this signal now, and then they will just record the number, like when you can hear this, and then they just draw this graph, okay, based on this practical experiment with several participants. Yeah, they bring in. So this is did like in a lab. So it's not like really it's not math, right? It's not theoretically derived, but they did this like experimentally or practically with real humans. And there will be some errors probably, and also some discrepancy between different participants. So this should be some average, okay, some average curve, which gives you a rough sense of how this auditory masking works. Okay, so, and this is how they kind of test on how they determine the bandpath of these auditory filters, and this is also how people realize, right, there exists the auditory filter, which kind of has this a band range, right, a band limit, a band range there. which means you mask not only the exact same frequency, but also, you know, frequency near that thing will also be masked heavily. However, this is auditory masking in frequency domain, but also there are also temporal masking in time domain, which means, like, for example, I'm talking now and you are playing something that's kind of simultaneously happening with my speech, and then my talk will definitely, you know, be masked by that sound if that sound is of the is in this within this, you know, band limit with my speech. However, the interesting thing is that it's not only the simultaneous masking is happening, and there's also pre-masking and post-masking. So I think post-masking might be easier for you to understand. For example, you hear a very loud bomb, like, you know, the bomb, or a very loud sound. And after that, for maybe a few milliseconds, you know, you basically cannot hear any other things. So this is a post-masking. So even if that sound kind of terminated, but your auditory system doesn't really, you know, is not accustomed to this sound, because this is very, you know, very sudden and very striking. And this kind of sound will not only simultaneously masking, you know, what else, for other kind of sound sources. After the sound terminates, it will also post-masking the sound source, like for like, you know, a few milliseconds, not really a few, like already 100 milliseconds, right? So like 0.3, 0.5, or right. Oh no, it's really 0.1, 0.2, right? 0.1 seconds or 0.2 seconds. So it's a very short time, not really very long. But I mean, it depends on the sound level, right? The loudness of the sound. If the sound is really loud, and it will also possible to damage your auditory processing, right? Which cause you to be deaf, and you won't hear anything in the future. But we're just saying this sound is within this normal range that won't damage your hearing. But when it happens, it will also have some post effect on your auditory system, which will mask other sound source for, you know, a few kind of, you know, 0.1 seconds, 0.2 seconds, like that. But there is also another interesting, there is also called pre-masking. So me personally, I don't really kind of understand this, but based on some research, okay, experiment by the scientists, they also discovered there will be also pre-masking. So pre-masking means before this sound happens, there will be a very short period of time you also cannot hear anything. So this is before the sound happens. For this, why it happens, I'm not sure. I really, because I'm not an expert in this field, in this kind of, you know, physiology kind of things. But yeah, based on their experiment and research, they showed that there is also some pre-masking effect before a sound, you know, occurs. So right before that sound occurs, for maybe a few milliseconds, you also cannot hear, you know, other sound. It will also mask other auditory objects. So this is the kind of graph, they did in the lab, and kind of a thing that you know, shows how the temporal masking works for our auditory systems. And from auditory filters, people derive something called critical band. So critical band is actually the same thing or similar thing as auditory filters, but critical band is more used, you know, in this engineering or not that biological, you know, context. So it's a different terminology, which will mostly be used, you know, by people doing, you know, who are not who don't work on this physiology, you know, physiology, biology field, whatever. So in audiology and the psychoacoustics, the concept of critical bands introduced by Harvey Fletcher describes the frequency bandwidth of the auditory filter created by the cochlea. roughly, the critical band is the band of audio frequencies within which a second tune will interfere with the perception of the first tune by auditory masking. So it will cause the beating and auditory roughness. Okay, so we talk about beating. So we play these two sine waves of very close frequencies. Like we use 400 hertz, and if we do this 401 or 402 hertz, they will cause this beating, right? Beating effect. So, and this will also cause some auditory roughness, makes this, two kind of sounds, but we actually perceive them as the same kind of source. If you remember beating, right? You don't really hear 400 and 401 hertz separately. So they are kind of interfering with each other. They fuse, right? It's all called auditory fusion. They fuse with each other, which cause this beating effect and auditory roughness, but we don't perceive them as different frequencies, as two frequencies. However, if we play something like 300 hertz and 400 hertz, then for I mean, maybe some people also don't distinguish them, but for trained ears, it's comparatively easy to distinguish these two different frequencies. So I can tell that even if they happen simultaneously, I can tell there is a 400 hertz and a 300 hertz separately. But for two frequencies happening in the same critical band, it's very difficult, for us to distinguish two different frequencies. So they will fuse with each other, they will interfere, and they will cause a kind of auditory phenomenon, but they are kind of perceived as one. Okay, so they are nonlinear, level dependent, and the bandwidth decreases from the base to apex of the cochlea as the tuning on the basilar membrane changes from high to low frequency. So this describes the shape of the bandwidth. So this is helpful because we are going to talk about how to design those bandpass filters, right? Engineering, from this engineering perspective, because we want to kind of reflect the reality of our auditory system. This will also happen with many kind of speech analysis or speech generation kind of tasks. Even if you use neural network or any speech speech kind of regeneration or kind of avoid the distortion or whatever. I mean the task related to audio, speech, and music, etc. It's very helpful for us to understand how we really perceive sound, and then we can design a similar structure engineeringly that will save much effort and will have a more precise reflection of the sound representation for human. So actually the bandwidth, okay, they are non-linear, level dependent, and the decreases. Okay, it's not like the same bandwidth for every frequency. So for different frequencies, they will have different bandwidth. So normally the bandwidth decreases from the base to apex, which means it decreases from high to low frequencies. So in low frequencies, the bandwidth is smaller, but in high frequencies, the bandwidth is bigger. Okay, so we'll talk about this later on. We will so because of this kind of structure, then engineeringly in this DSP, right, digital audio processing, actually, so we talk about Fourier transform, right? But Fourier transform actually treat all the frequencies the same. So we'll do Fourier transform for all the frequencies. You kind of use the same bandwidth for all frequencies, from 0 to whatever. This is Fourier transform. So they are equally divided. Okay, in the frequency domain, they are equally divided. But we know that in our real auditory system, those bandwidth are different. They are frequency dependent. Therefore, for the engineer, they devised another method another kind of mechanism which is called a male frequency spectrum or male spectrogram. So this male spectrogram is actually a built-in function in MATLAB, and this is algorithm. Okay, so you can see you have some buffer window, and you do this FFT fully transform, but actually they use this kind of male filter bank. Okay, so what is a male filter bank? It looks like this. Okay, so you can see this instead of equally or dividing the frequency range to equal distance, like you have some unit like 100, 200, 300, 400. So this will be equal distance. Instead of doing that, we want to simulate more of the real human auditory system. Then they designed this male spectrum or male filter bank, where in the low frequencies they have smaller or narrower bandwidth, and when the frequency increases, in the high frequencies we will have wider bandwidth. Okay, so this is the kind of, but it's also an approximation. I mean, we need to realize that our real auditory band doesn't look exactly like this. Okay, this is also a kind of engineering approximation, but comparing to fully transform, it better reflects our real perception. Okay, because it incorporates this the bandwidth of auditory filters into the DSP tasks. Okay. So now I think we need to talk about what is male scale. Okay.